central dogma of molecular biology. So you will be hearing this terminology. Dogma அப்படின்னா என்னது? கோட்பாடு, right? கோட்பாடு, like or a rule. So central dogma of molecular biology. So this central dogma is how all this entire human being or individual is made from the information that we cannot read, right? So how the system reads this information. So all this. So if it needs to make me, right? And so there needs to be information in my DNA. So all that DNA. So in each and every part of the cell. But that information, so that information should specify where the my eye should be, right? கண்ணு எங்க வரணும் அப்படின்னு it specifies. So if that information is not there, I can have eyes anywhere in my body. I will show you some examples, right? So this eyes can be anywhere in the body if we alter that change, the position of those genes, right? This is called as homeotic genes. So how this information is read by the system. So we need to know that information, right? We cannot read. So like you can read that because you know the alphabets, right? So you know from the UKG, LKG, UKG, you have read you have 26 alphabets in say like English, right? You put together those alphabets, right? 26 different alphabets you put together and you get each of those words, right? So you have each word. So each word, each word will be the codon, right? So all the central, so this alone will not give you any information, right? So many number of these codons attached together will form a sentence, right? It will give a sentence, but each this sentence will not give you a detailed information. So all this sentence or 3 or 4 sentences put together will give you one paragraph which will give you some information. So you need to have like words, sentences, paragraph and a chapter which will give you the structure. Right? To understand what the DNA, we need to understand the DNA language. Right? So DNA has only 4 alphabets, ATGC. So how these alphabets are put together to form the information. So this central dogma of molecular biology was given by with this Crick, right? Francis Crick. So he put forth the central dogma of molecular biology, where this flow of genetic information occurs. So first, the DNA can replicate onto itself, right? And the DNA by transcription make a copy of the gene, which will be the RNA, right? So this RNA will be translated translated into a protein, which can give rise to a specific trait, right? So this is the central dogma given by Crick. Now this has they are modified a little bit, right? We will see the final central dogma. This model was given by Crick. So the messages. So why do we need to know what the DNA actually gives the message? So you can read this because this consists of English alphabets, right? So what does it say? Can you read it? Molecular biology is, right? Because you know the English alphabets, you can put together and read something. No. Anybody know music? So what is read? So this can be read only by people who knows the music alphabets, right? Right. So we need to know the alphabets of this. And you generation, you may not have seen this. So this is the Morse code, right? So in our times there was telegram, not your app telegram. So how did you get that app telegram app? So that was given in the Morse code, like dots and dashes. So this make up each of those alphabets and also words, right? So when we understand what each of those alphabet means, then we can get the entire information, right? So now we have this. Again, we don't know this alphabet, so we cannot read it. So we need to understand what each of those alphabets is giving rise to each of those words, how to make up words and how the words make up a sentence and that sentence to a paragraph. So each gene, we need to understand what each codon makes up and what protein it produces. Now this is the language of DNA, right? So now we need to understand what each of those nucleotides specifies and what this information, how this is being translated into a structural unit, right? So the DNA transforms the specifies the sequence of bases along the DNA strands, and the DNA leads to specific traits by indicating by dictating the synthesis of specific proteins, how much to produce, where to produce, right? Such type of information. All those information is present in the DNA, right? How much to produce, where to produce, like we have seen mitochondrial proteins. So that will be synthesized in the cytosol. That will have information for mitochondrial signaling. So that will be transmitted or taken up to the mitochondria or chloroplast. So each specific, so protein synthesis, where it should go, how much to produce, all the information is in the DNA. So like the proteins or the links between genotype and the phenotype. All right, the examples like you have already seen from even the 10th standard, you have the Mendel's pea plants, like the dwarf on the tall or a smooth wrinkled piece. So how this arises? How this arises? So this in the dwarf piece. So the Mendel dwarf P lacks a functional copy of the gene that specifies the synthesis of a key protein called as the gibberellins. Right. So gibberellin is the hormone responsible for elongation of stems. So when that gets mutated, just a single gene gets mutated, you will get a dwarf plant. Right. So gibberellin stimulate the normal elongation of stems. Say like the plant height. So you have the tall and the dwarf plant. But when we look at this, the dwarf plant, it will have all the internodes as that of the tall plants. Tall plants எவ்வளவு nodes இருக்கும். The nodes are where the leaves arises, right? So இப்போ இங்க 1, 2, 3, 4, 5 இருந்துச்சுன்னா இங்கேயும் 5 nodes இருக்கும். But it will be dwarf, right? So why this does not elongate? Because of the gene gibberellin 3 beta hydroxylase, right? So when this actually the normal gibberellin hydroxylase adds a hydroxyl group to GA20 and makes it a functional copy, right? So this is GA20 அப்படின்னா this is 20 times less active than this, right? So when there is a mutation in GA1, this is 20 times, this 20 times less active. Right, so this regulates the cell division and also the elongation, and a single nucleotide mutation converts the alanine to 309. So one amino acid, alanine, is converted to 309, which in the final protein, the mutant protein is only 1/20th active as the normal GA. Right, so this entire plant will be dwarfed. Right. So another one, the seed shape. Seed shape, you have the smooth piece and the wrinkled piece, right? So have you differentiated between the smooth and the wrinkled piece? Have you eaten peas? பட்டாணி சாப்பிட்டிருக்கீங்களா இல்லையா? Right. Yes, right, okay. So have you differentiated between the smooth and the wrinkled piece? அவ்வளவுதான். So usually when you eat the piece, those wrinkled piece will be much more like 10 times more sweeter than the round piece, right? This will be much more sweeter than this round piece. Why? So this gene, well, which is called as the starch branching enzyme, SBE1, starch branching enzyme. So starch is just the sucrose molecules. Right, glucose sucrose molecules, where when combined together it will form a branches like beta branches and alpha, beta and alpha branching, so that give rise to the starch, right? So the normally when the seeds are filled, so it will be the glucose that will be converted into starch, and if the starch branching enzyme is mutant, then it cannot add more branches to this glucose or sucrose, right? So when the starch is not present, so only the glucose will be present, right? So when the seeds dry up, so the starch will be very hard when it dries up. So that will be a smooth seed. But the glucose or sucrose, when it dries up, it is not hard. So that will be wrinkled. So that will also be sweet, much more than this one, right? So a single starch branching enzyme mutation. So the function is it adds change to the charge, and because of transport insertion into inside the gene, because of the mutation, the SBE activity produces less starch and more of sucrose and more of water. After drying, a seed loses more water and it gets wrinkled. Right. So this single gene mutation can give rise to either of those phenotypes. Hmm. So the gene concepts. So one gene, one polypeptide, which was given by Beadle and Tatum, right? So they experimented on the fungus Neurospora crassa. So how they came to put forth that one gene is responsible for one enzyme, right? So, what is it? Okay, so right. So this Beadle and Totem has to establish the link between the enzyme and the genes, and they studied on the bread mold, the fungus, Neurospora crassa, and they gave this one gene, one enzyme hypothesis. So based on this, there is each of this. So what's the use of studying this mutants? And the mutants in the sense, mutant is something lacking or present that's not present in the normal individual, right? So mutant can be additive or negative, right? Negative is not present. Additive is present more than the normal individual, right? So how can we study the mutant in the sense like how they studied, or like for example, us say like how do we know the function of hand, right? Because it can grab something. So if all of you don't have two hands, right? Or all of you don't have two hands, but I have two hands, right? So எனக்கு மாத்திரம் இருந்துச்சுன்னா, I am a mutant from you, from the normal population. So how will you know the function of this hands? Because I can show you that I can grab this, I can put food in my mouth. So I can show you some function that is responsible by this unit, right? So if I am not having this and you are having this, and you will know that the lack of this will make up this lack of some other function, right? So lack of one gene can give rise to the presence or absence of one character, right? So what they studied is mutate one gene, mutate the entire organism and find out which gene is responsible for which character, right? Because we have 30,000 genes. We don't know which gene is responsible for which character. So how can we study that? By mutation, right? So here, so you have the neurospora produces using the spores, right? So when we mutagenize using the X-rays, right? So X-rays, some spores have random mutations, and if you put it on the media, right? So all of this spores can germinate and give rise to transfer each of the spore to its own complete media first. So complete media has all the amino acids required, right? So if it has even if it is mutated, if it has all the amino acids present, then it will grow, right? Because it need not synthesize. So neurospora needs to have enzymes for the synthesis of all the amino acids. So if it cannot synthesize, right? So we need to supplement from others, right? That is be the essential, non-essential amino acids in biochemistry, what you have read, right? No, non-essential amino acids are one, you can synthesize, right? Essential amino acids, you need to get it from outside, right? So that if you give everything from outside, then even if it is mutated, then it can grow, right? So this transfer part of this each colony onto a nutritional mutant, right? So or deficient. So this nutrition mutant or when on the minimal media. So when you transfer each of those cultures into a minimal is deficient in any of those amino acids, right? So then that colony cannot grow. So and also, so in each of those media have 20 different amino acids, right? So there's glycine, phenylalanine, methionine, arginine, cytosine, lysine. Only this amino acid present, right? Only present or absent. So transfer each of those colonies to all of those 20 tubes. So if this colony can produce all other amino acid except arginine, right? So then it cannot grow in all of the any of those amino acid supplemented media. But because this cannot synthesize arginine, right? So that will be growing in the media containing only the arginine, right? So when going back, so we have several of those pores, right? So each of those pores or each individual when it gets mutated. So this colony got mutated in only one amino acid. So one gene when gets mutated, the gets say like mutated with only one amino acid requirement. So one gene is responsible for one amino acid synthesis. So there can also be possibility that two or three different genes got mutated and it can grow in presence of two or three different. That will be multiple genes. So what they got is one gene that got mutated that can grow in only one presence of amino acid. That is the only lucky one. Just like Gregor Mendel, whatever the character he has chosen only one gene mutation characters, like the plant height or say like seed, or the flower color. These are all single gene characters. If he has taken leaf shape, those are all polygenic characters, right? So then there will be many of those combinations and he will not be lucky. So here they were lucky that one gene got mutated for each amino acid so that they can find out each gene is responsible for one amino acid. One gene, one enzyme hypothesis, and this gave rise to the one gene, one protein, right? One protein, and later research that many proteins are composed of several polypeptides, so that each gene can give rise to one polypeptide, not just functional units, can also be a structural unit of proteins. So this central dogma of molecular biology, so where the DNA gets transcribed to RNA inside the nucleus, right? Inside the nucleus makes mRNA, makes mature RNA. So where there will be lots of say like processing in the RNA also, right? So that mature RNA will be exported into the cytosol where it will be translated into the protein. So now the central dogma of molecular biology has changed from DNA can replicate onto itself. So now this red ones are the latest ones, right? What we know. So the DNA can transcribe into RNA, and also RNA can also be reverse transcribed to DNA, right? And RNA can be translated into protein, and this protein but cannot go back to either RNA or DNA, right? So this is not possible. And also protein cannot also not able to replicate itself, will not be able to replicate itself, or the protein also cannot go back to DNA. So the Francis given by Francis Crick. So this can be in 1 or 2 mark question. Central dogma given by Dash or Whom. So central dogma of molecular biology deals with the residue by residue transfer of sequential information. States that such information cannot be transferred back from protein to either protein or to the nucleic acids. Protein cannot go back to the nucleic acid or RNA. So protein information cannot flow back to nucleic acids, and the fundamental framework to the understanding the transfer of sequence information between all this biopolymers. The transcription and translation are the two main processes linking to the gene protein, and the genes provide all the instructions to make the specific proteins, and the bridge between the DNA and the protein synthesis will be the temporary RNAs. And RNA is chemically similar to DNA, except that it contains ribose as its sugar and substitutes the uracil for thymine. And RNA molecules are always single-stranded. The RNA has four monomers, and it has specific sequence of hundreds or thousands of nucleic acids, and the primary structure of protein, and it's written in one chemical language which requires the transcription and translation of the DNA to the protein. So during transcription, the DNA provides template for the complementary RNA synthesis, and this RNA gives rise, and it is read the transcription is from the copying is from 3 prime to 5 prime. The reading will be 5 prime to 3 prime direction. So this transcription produces mRNA, which carries the message from the nucleus to the ribosomes for translation. So the basic cells, so depending upon how this is being organized, so the prokaryotes has the DNA suspended in the cytoplasm. There is no specific nucleus, but here the DNA is inside the nucleus. So here, so the difference, the basic mechanism, the difference between the prokaryotes and the eukaryotes is that the bacteria lacks nuclei, and the transcription and the translation are also coupled inside, which takes place inside the cytoplasm. And the ribosomes attached leads to the mRNA, and the transcription and translation are still in progress. Like both transcription, translation, everything happens within the cytoplasm, right, in the bacteria. So even while it is being transcribed, it can be translated. You will see that in translation in prokaryotes. But here in the eukaryotes, so in the prokaryotic cell, even when it is being transcribed, the mRNA, that will be again translated. But in the eukaryotes, so first the mRNA needs to be synthesized. It needs to be processed into the pre-mRNA and mature mRNA, and it will be exported in the cytoplasm, where it will be translated by the ribosomes in the cytoplasm, right? So this the transcription and the translation are uncoupled in the eukaryotes, but here in the prokaryotes, both are simultaneous. So now the flow of information, now, what we know now is that so the DNA can be replicated. DNA to do a DNA by DNA polymerase. It can replicate onto itself. The DNA can replicate onto itself, and by transcription, the DNA is converted into RNA by RNA polymerases into single-stranded RNA, and also by reverse transcriptase. So the RNA can be converted into DNA, right, by reverse transcription. And this mRNA can also be translated the RNA to protein by the ribosomes to give rise to an entire protein. Or it can also be the RNA can also be replicated into another RNA, right? Also called as miRNA, microRNAs, or short RNAs, right? So this can be like the positive RNA, sense RNA, or negative sense RNA, right? So this will be the RNAi, interfering RNAs. So this has applicability in genetic engineering. So now when you see this, so this is plus RNA, this is minus RNA, right? So what can happen? So this RNA can bind to this, right? This can complement this. So RNA one the single stranded. We know that RNA is single stranded. Why is it single stranded? So for ease of use, right? If there is double stranded RNA in the system, right? So the cells will degrade that immediately, right? Because viruses, so that is one of those immune systems. So viruses has RNA. When it injects its RNA, so that can go and bind to our system. So if the system recognizes double-stranded RNA, it will immediately cleave that, right? So when we have this negative sense RNA, right? So this can go and bind to this positive sense RNA. That will be a double-stranded. So immediately the system will cleave all that double-stranded RNA and reduce the RNA. So this has applicability in fruit ripening and other genetic engineering mechanisms. So the transcription, so it transcribes the RNA into this sense RNAs. We'll see all those. On the types of RNA, you have this messenger RNA that codes for the entire actual coding template. Then transfer RNA, these are the RNAs that brings, they recognize the encoded sequences and then brings in the amino acid chain for the polypeptide chain, right? So this the cloverleaf model. We will see in detail all of this. So this has the anticodon end. So the anticodon end which recognizes the actual. So this will be the codon on the mRNA, like the triplet. So that recognizes what is present in the codon, which will be the anticodon corresponding the amino acid, right? And the ribosomal RNA, this forms the ribosomes, to form the structure of the ribosomes. The proteins catalyze all the function of the polypeptide synthesis. So each of the ribosome is also made up of this ribosomal RNAs. So during translation, so all this the tRNA will recognize the ribosome binds the small subunit and the larger subunit binds to this mRNA, and the tRNA brings in the amino acids, and then the polypeptide chain. Polypeptide chain will grow onto the mRNA. So this will be the final what we have seen. DNA produces RNA, and RNA produces protein through translation and different forms. Summarize DNA to RNA, which will be called as the central dogma of molecular biology.